Okay, moving on to presentations and discussions. I'd like to invite Dalena Garcia-Sosa to come and join us. Hello. Hi.
My name's Dalena Garcia-Sosa. I started volunteering with Bari Mitchell 2 years ago, and in the process, working with Val, we found something pretty interesting in our research, and I just wanted to tell you guys everything that we found so far. To give you a little bit of an introduction, here's the anatomy of a bay scallop. What we specifically study the gonad. They're hermaphrodites, so they both have the female and the male. And what we found was a parasite in the intestines, and as you can see, the intestines runs through the gonad, so it seemed to have some type of effect on their reproductive system. So about the last day when I was working with Val, Dr. Howe, we found something in the intestines that was not supposed to be there, and it seemed to have some type of membrane around it, and it seemed to be like an actual alive thing. And Val had never seen anything like it before, so we thought it was a parasite. And so she was retiring, and I thought it was really unfair that we weren't going to be able to continue it. So I asked around the school. I talked to my marine science teacher, Kara, and Miss Vassal and everything, and they helped me organize my own independent study to help me continue on Val's research. In the process, I made 3 hypotheses with the information I had at the time, which was, of course, there's a new parasite in the scallop intestines. I considered rises in temperature that may be causing them to survive, and also that combined with the rises in temperature and the presence of the parasites, it causes the eggs to become atresia. And this basically means that the eggs basically die out and they don't work anymore for reproduction. You can see how they're supposed to look and what it looks when they're atresia. I used the method of histology that Val taught me, which was collecting, dissecting, then preparing them for embedding, and then sectioning them and staining and a microscopy. Some pictures. The first one's of me dissecting, second one is collecting, and the last one's trimming the blocks to get ready for sectioning. This is the data I found. We collected 5 scallops from different parts of the harbor, and I counted how many had the parasite and how many didn't have anything wrong with them, and then I calculated the average of them and the percentage of parasites that were out of all the scallops that I collected. Here's a little graph. As you can see, the only one that had not as many parasites as the rest was First Bend inshore, which had a slightly higher temperature than the rest of the sites. And out of all of the data I collected, I concluded that the only hypothesis I got correct was the first one. Even though the parasite's present, it doesn't seem like the rises in temperature really affected, as you can see. And the specimen we collected were too young to be able to determine whether the eggs were atresia or not, so I kind of crossed that off the list. Some ways to collect better results, more accurate ones, was get a better sample size, collect scallops no matter how old they are, and study the digestive tract itself instead of the gonad in the sexual reproductive system. How can you help? 2 discussion questions that you guys can talk about is how this can affect the scalloping industry, because as we all know, scalloping is very important for Nantucket's economy and overall the culinary industry around the world. Nantucket-based scallops are pretty popular. And overall, what's the real effect of the parasite, not just on the scallop, but the environment itself. Scallops are filter feeders, so they basically clean out the water and the harbor themselves when they eat. And I wanted to present an idea for Nantucket High School to work together with Mariah Mitchell and Egan Maritime, if possible, to create a program for students that are interested in marine biology, marine science, and any type of life science to help me and everyone else involved to continue my research and help solve everything. Program could be in the fall, I can help and teach all the students that would be participating in the process since, Val's retired. And it could motivate a lot of students to follow their dreams. I've wanted to be a marine biologist since like 3rd grade, and the fact that I got this amazing opportunity helped motivate me so much and helped me believe that like it was actually possible for me to do this and get my career. And it can also just spread awareness around the environment themselves, how the harbor's doing, and how the whole island can be affected by this. Some special thanks. Dr. Valhal, she's in the audience. Jordan Bond, who helped me, who drove me to the lab and to the hatchery, and if it wasn't for him, I wouldn't get a ride anywhere. The hatchery and Tara Riley, the leader, they helped me with microscopy, and I would go over there, use their microscope, and they helped me with collections. Janelle Gurley and Mariah Mitchell, I used their lab, and they were overall a great help. Mandy Bassel, I hope you don't mind calling you Mandy. She supported me a lot, and she helped me organize everything, and she allowed me to take class time and time out of my school day to go to the lab and help. Kara, my marine science teacher, who started basically all of this and talked to everyone and helped me organize everything. And lastly, Heather McDonald, my bio teacher, taught me honors bio, and now she's my AP bio teacher. She's amazing, like probably my favorite teacher I've ever had, and that's it.
Great. Thank you. I'd like to just see if the committee has any questions
Yes,
or
of
comments
course.
for you.
Yeah.
Tim?
I think that's wonderful.
Thank you.
I worked in a lab with human tissue, but doing the same things you were doing, and it's very interesting stuff, and I think that's amazing.
Thank
it's really
you very
good.
much. Thank you very much. Any other questions?
Chantal?
What year are you in school?
I'm a sophomore,
Okay,
incoming
good.
junior.
Okay. Just making sure you still have time to continue.
Yes, of course. I really hope I do. Anything else?
Esmeralda?
No questions, just great presentation.
Thank you very much.
Laura?
I echo Esmeralda's comments. You're so poised. This was really interesting, and I agree with Dr. Lepre. It is a wonderful program, and I would love to see this continue.
Thank you very much.
Yeah.
Thank you for the opportunity to present everything to you guys. If there's anything
Well, I
else.
just wanted to say I'm so glad that you have an opportunity to work with Dr. Hall before she completely retires, because she has been a gem in our community for years, and her passion for educating and passing on her knowledge to the next generation has been something she's done for a long time. And I am so impressed by you. As Laura said, you're so poised, and thank you for making a scientific report one that I could understand.
To be honest, the whole thing, it brought me a lot more questions than answers. I didn't really was able to discover as much, but I did find out at least a little bit more. So
I think Dr. Hallett has a
I do.
comment.
First of all, I'm so proud that you were able to do something like this. It's such a special thing. When I was a child, I wanted to be a marine biologist myself. That was what do you want to be when you grow up? It was marine biologist. I never got there, but that's okay. 2 questions, technical questions. Do you know if this parasite is a new species, or is it a species that exists already?
as of right now, me and Val believe that it is a completely new species. We went, she's never seen anything like it before, and we looked across articles about all different types of shellfish parasites, and we didn't find anything that looked like it. Or maybe it looked like it, but it wasn't specifically in the digestive tract. So as of right now, it seems like something completely new.
Wow, that sounds like possible naming rights. That's pretty cool. That's amazing. So I won't even ask, but I imagine this might be a next step for study, is wondering if it has any effect on humans.
Yeah, definitely, especially because people love eating them.
Of course. And then my last question, or not question, but statement is, I would love to support a program, perhaps it's an independent study, and I'm happy to work with Ms. Vassal to see if we can promote this and continue this for you and for other students who might be interested. We have such a resource at our fingertips with not only with Mariah Mitchell and Linda Loring and our other science organizations here on the island, but with the opportunities that we have through the rich environmental ecology opportunities we have right here. So I would love to see this continue. So hopefully we can talk about that soon.
Thank you very much. I know a lot of students myself, like personally, that are extremely interested and want to be like shark researchers, coral reef restorative like scientists. So that'd be great.
Thank you.
Thank you so
Thank you.
much.